So we have introduction to microbiology. Okay, microorganisms are microscopic organisms. Okay, these may be unicellular or multicellular. Okay, microorganisms. Body plan, they have single cell or cluster of cells, or organelles, they lack organelles. Size, dimension of microscopic organisms fall within the range of micrometers, sometimes nanometers. Most microbes extend from the smallest viruses to the much bigger protozoans. Smallest are viruses and bigger protozoans. Other habitat, they are ubiquitous. That means they are found any or everywhere. Yeah. Classification, they are generally classified as archaea, bacteria, fungi, viruses, protozoa, algae, right? Microorganisms to environment. Microorganisms such as algae, they produce oxygen. They decompose organic material, provide nutrients from plants. Microorganisms to humans. Some microorganisms are good for human health and some are pathogenic. Some are pathogenic. Factors affecting growth: nutrients, oxygen, water, temperature, acidity, light, and chemicals affect the growth of microorganisms. Next, we have study of the, so bacteriology is study of bacteria. Mycology is study of fungi, right? Protozoology is a study of protozoa. Then we have phycology, the study of algae. Okay, then we have parasitology. It is study of parasites. We have virology, the study of viruses. We have nematology, the study of nematodes. Next we have categories of microorganism. Okay, depending on this cell type, are divided into two categories, and I'm going to do the flow chart. It is eukaryotic and prokaryotic. Okay. Eukaryotic have complex cellular structure like humans and animals. Their cells have nuclei and mitochondria. Okay. And examples in eukaryotic we have examples fungi, helminthes, protozoa, and unicellular algae. In fungi we have molds and yeast. Helminthes have tapeworm, hookworms. Protozoa we have plasmodium. Unicellular algae we have Chlamydomonas. Okay, prokaryotic are simple, self-sufficient, having primitive nuclei, means old. Then we have mitochondria, which is capable of leading independent lives also. In prokaryotic, we have bacteria as example, which is Vibrio cholera, E. coli. Then in archaebacteria, we have Thermobacillus sps. Then in cyanobacteria, we have Spirulina sps. Then we have non-cellular microorganisms which are without any cell forms and are called as acellular. And also non-cellular. Okay. Viruses. Talking about viruses, they consist of DNA or RNA and proteins. They are not capable of leading independent life. They grow and multiply by infecting cells. So, higher organism. Viruses are not independent. Alright. Next we have prions. Prions are made up of only protein that cause bovine spongiform encephalopathy. Prions cause BSE, which is bovine spongiform encephalopathy and Cooney. It is a disease, etc. Okay. Then next we have virusoids. Virusoids are made up of single-stranded RNA and viroids are made up of circular but single-stranded RNA without protein. Okay, it causes viral hepatitis D. Then we have history of microbiology. It was not very clear that microscopic living organism exist. In 1600 AD, Anton van Leeuwenhoek built his own microscopes and made first descriptions of microscopic living creatures. Leeuwenhoek observed a variety of things like rain, water, pond water, and scrapings from his own teeth under the lens of his so-called microscope. He saw minute moving objects and called them as little animalcules, such as protozoa, yeast, and bacteria. He made accurate sketches and communicated his findings to the Royal Society of London. He had most likely observed single-celled eukaryotic microbes. He observed eukaryotic microbes. With this observation, the science of microbiology was started. For this reason, Anton Mon is called as the father of microbiology. Father of microbiology, it is Anton Mon. And talking about Edward Jenner, Jenner was an English doctor, okay, who discovered vaccination against smallpox. Vaccination against smallpox. This ultimately led to eradication of smallpox. He observed that dairy workers exposed to occupational cowpox infection were naturally immune to smallpox. In 1796, he experimentally proved that resistance to smallpox can be induced by injecting cowpox material from diseased pustules into man. By injecting cowpox material from disease into man. Jenner published his findings in 1798 in a pamphlet which is an inquiry into the cause and effect of variole vaccine. Our next is the Louis Pasteur. Okay, he was a professor of chemistry at the University of Lille, France. He is also called as the father of modern microbiology. Generation experimentally by using swan-necked flowers experiments. He experimentally showed that soaring of iron and bear is due to the growth of undesirable organism. He also showed that the desirable microorganism produce alcohol by a chemical process called fermentation. Alright, he introduced the technique of pasteurization and proposed a germ theory of disease. This theory was developed in 1860 by Louis Pasteur. He stated that tiny living beings are the cause of infectious diseases. While doing his research in puerperal fever or childbirth fever, Louis Pasteur proposed this theory. This disease for the first time supposed that pathogenic microorganisms such as bacteria and viruses could cause diseases. The term germ here stands for microscopic organism that cause infectious diseases. We have pasteurization. It is a special technique of heat sterilization to kill germs. Alright, It is used in milk now. It includes heating the milk from 60 to 65 degrees Celsius for 30 minutes, followed by rapid cooling. The decrease of rapid curdling of milk due to the action of microorganisms. Louis also developed steam sterilization technique. Today popularly called as autoclaving. He experimented differentiated between aerobic and anaerobic bacteria and coined the term anaerobic. Refer to organisms that do not require oxygen for growth. He developed anthrax vaccine. Yeah. Sorry. He also developed a vaccine against rabies, which is hydrophobia. Vaccine against rabies. Okay. Then we have Robert Koch or Robert Koch. It was from 1843 to 1912. He was a German doctor who later became the professor of hygiene and director of institute. Was a German doctor. Okay. He's known as father of practical bacteriology. Practical bacteriology. He's also regarded as the father of medical microbiology and bacteriology. He discovered the Bacillus anthracis. Anthracis. Okay, the casual organism or disease of anthrax. He introduced staining techniques for the study of disease causing microorganisms. Staining techniques. He discovered Mycobacterium tuberculosis, which causes tuberculosis in human beings. He discovered Vibrio cholera also, or Vibrio cholera, whatever, which is a causative agent of cholera disease. He developed pure culture techniques by introducing solid media. He had a great contribution to development of pure culture technique. He records postulates. And we have Joseph Lister. He was from 1827 to 1912. Professor of surgery. As well as the father of antiseptic surgery. First person to use an antiseptic solution during the process of surgery. He used carbolic acid, which is phenol, on the wound during surgery, which successfully prevented sepsis. He introduced application of carbolic acid during wound dressing. So, antiseptic surgery later led to development of aseptic surgery. We have Paul Ehrlich also. 1854 to 1915. He was a German bacteriologist. He used a technique of chemotherapy in medicine. He is called as the father of chemotherapy. The Vedic concept of chemotherapy is based on the fact that organism causing disease could selectively be killed with chemical drugs without harming the host. He produced first synthetic drug, which was Earth's phenamine to control the syphilis disease. Syphilis disease. Alright. He also observed that drug would undergo certain changes in the body after it would produce a desired reaction.